Okay, so this is the 7th session, TA session for biointerface engineering. And today we'll cover the week 6 assignment. Okay, so these were the topics that were covered for week 6 and the week 7. So in week 6, host response to biomaterials, cell adhesion, and biocompatibility of the biomaterials was discussed. Wherein the cell surface interactions, complement pathways, the sequence of responses were discussed, and also different mechanism of cell adhesion, the cell adhesion molecules, etc. And in the third lectures, that is biocompatibility of biomaterials, the techniques to assess biocompatibility was also discussed. And for week 6, surface modification was the focus. So surface modification uh, and the objectives of surface modification, the different techniques and the different techniques, physical, chemical and biological, were all discussed. And in the third lecture, a specific case of coating of calcium phosphates on titanium implant was discussed. So we'll start with the assignment now. So this was the first question. Before we start, Sakshi, were you able to uh, raise a concern regarding the question in the previous assignment?
Actually, I had put a mail on the discussion forum, but I think I haven't received the reply from such a thing. Like, is there anything from your end that you can do?
I don't have contact with the TAs that are handling these things, the technical things. So I'm not sure, but I'll try to find out if I can.
Sure, sure.
Okay. So the first question was, which of the following sequence of events occur during the cell surface interactions? And the events are A, cells, B, surface water, and C, surface protein. And here the correct answer would be PCA, that is option 3. So the surface comes in contact, as soon as the surface is exposed to the body, first the water comes in interactions with the surface, that is followed by protein, and these protein then attract cells. So the sequence is B, C, and A. Okay, so this was the next question. Fibrins are degraded by what? So here the correct answer would be thrombin. So this happens during the homeostasis, the first step of wound healing. So during that step, thrombin, which is an enzyme, it degrades fibrinogen into fibrin and fibrinogen, and it leads to fibrin. So basically, the thrombin acts on this fibrinopeptides, and that removes this fibrinopeptides, exposing the binding sites. So these fibrin monomers can then bind together to form insoluble fibrin or the fibrin plug, the final clot. Okay. So the next question was, which of the following is or are involved in cell adhesion? The options were focal adhesion, extracellular matrix contact, and close adhesion. So here, all of these would be the correct answers. And as shown in the lecture also, these three are involved in cell adhesion. Which of the following does not belong to the class of cell adhesion molecules? So the options were cadherins, albumin, immunoglobulin G, and mucin-like molecules. So the correct answer here would be albumin, and the rest of the options, that is cadherin, immunoglobulin G, and mucin-like molecules, as well as integrins, these are all part of the cell adhesion molecules. So the correct answer would be the second option. Now this was the next question. The most common amino acid sequence RGD bind to which cell adhesion molecule? Cadherins, integrins, selectins, or immunoglobulin superfamily. So here the correct answer would be integrins. So RGDs have these particular sequence, that is arginine, glycine, and aspartic acid, and then the it reacts. Integrins have specific RGD receptors, so the RGD binds to these integrins via these ligands. Here is the next question. The percentage of cell adhesion over the surface can be estimated by relating the available surface area for cell adhesion, initial cell number available for cell adhesion, and the rate of cell adhesion. So here the correct answer would be all of these. And there is nothing much to explain here. So it's pretty straightforward from the options. All of these can be used to measure the cell adhesion, percentage of cell adhesion. Now this was the next question. Which of the following criteria is not required for the biocompatibility of biomaterial? Options were cytotoxicity, leachability, hemocompatibility, and ductility. So here the correct answer is ductility, whereas cytotoxicity, leachability, and hematocompatibility are important in vitro assessments. Ductility is just a property of metal, so it is not specifically required to check the biocompatibility. The next question was, the in vivo analysis of the biomaterial implant involves. Options were histochemical analysis, immunohistochemical analysis, biochemical analysis, and all of the above. The correct answer is all of the above. And as shown in this particular slide, in vivo analysis involves both histology and histochemistry, immunohistochemistry, and imaging such as TEM and SEM, and biochemistry. So all of these are involved in vivo analysis. Can you still hear me, Sakshi?
Yeah, I'm there.
Okay. Okay, so the next question was, which of the following product is generated during the MPT assay? So the options were formazan, dimethyl sulfoxide, hetero, polymolybdenum blue, sodium molybdate. So the correct answer is formazan. So MPT is a dye, that is 3,4,5 dimethyl thiazole, 2,5 diphenyl tetrazolium bromide. This dye, upon action of the NADPH dependent mitochondrial dehydrogenase, they form purple formazan crystals, and which can then be detected by using absorbance. So here is the next question. Which of the following materials is not suitable for knee joint replacement implant? So here the options were scaffolds, polymers, metals, and composites. And the correct answer would be scaffolds. So polymers, metals, and composites, the orthopedics, basically orthopedics or joints, these are all related to the joint replacement implant. So metals, polymers, and other composites as well can be used for this knee joint replacement implants. Yeah, I think that's all I have for today. I couldn't prepare additional questions, sample questions for the next session. So we'll discuss the next session next week. If you have any doubts, or you you want to ask something else, then you can.
At present, I don't have any doubts.
Okay. Then do you want me to close this session?
Yeah, it's fine. I just have a little request regarding that assignment.
Sure, sure.
If
I'll
if
I'll
it could.
I'll see what I can do from my end.
Yeah. Thank you, ma'am.
Yeah. Thank you.
You can close the session.